Hello, everyone. My name is Madhavi Ganapathi. I'm a faculty at University of Pittsburgh, and I'm currently on a visiting position at Carnegie Mellon University, Qatar. My talk is about patterns like palindromes and inverted repeats in viral genomes. So what you get out of this talk is basically gain familiarity with what are DNA palindromes and inverted repeats, and understand the significance of DNA palindromes in genomes, and sort of look at whether they can be identified efficiently, computationally on a large scale, and also what did we find from our analysis, how common are these patterns in the genomes. DNA palindromes are similar to palindromes in languages. In language, in natural languages, basically the words will read the same forward and backwards. So you have race car, and it could also be a sentence where you say, it's Abel was I, or I saw Elba. So if you see the sentence, it reads the same forward and backwards. In fact, there is a book, Telugu book, in which there are poems that are written in this type of palindromic form. So Dhira Sayaniya Saradhi. So this, basically you get the point, these are palindromes. In DNA, on the other hand, we don't consider them reading the same forward and backwards. When you read it backwards, it's actually a complement of the forward sequence. So G complements with C, T complements with A, and so on and so forth. And this has significance in DNA palindromes. I actually want to point out one more thing. So if you have a DNA palindrome like this, and you compare, the DNA complement sequence, right? The second strand, if you read on the secondary strand, you'll come across a sequence that reads identical to what you have read over here. So the sequence in forward direction on the main strand will be the same as on the complementary strand when you read it from that direction. So these patterns are extremely important because they can play a role in the DNA stability and also various other mechanisms. So if you look at the regular palindromes without a break between them, they are actually responsible. I mean, they could play a role in DNA breakage and chromosomal translocation and so on, including the ones with the gap sequences. Sometimes even palindromes with a few mismatches could behave analogous to perfect palindromes. We were interested in studying this, and we carried out this analysis earlier, and we studied the DNA palindromes. That is where there is no gap sequence. We developed a tool in our toolkit to find all of those patterns. And then we went ahead and studied that on human genome sequence. But now the question is, first of all, what about inverted repeats, which means there is a gap between the two occurrences? And secondly, other than human genomes, what is their significance? So our collaborator brought this to, he informed us, educated us that this type of, they're called terminal repeats, inverted terminal repeats in viruses. And especially there's one in adeno-associated virus, which is very well known, that these type of terminal repeats are extremely crucial for the ability of the virus to infect the host cells. The question is, okay, we know about this. Are there other viruses that also have similar inverted repeats? So we went about studying this. And the idea is that these methods have to be scalable. They have to scale to long sequences, whether you study viral genomes or human genomes, and they also have to scale to tens or hundreds or thousands of genomes. In human, there are personal genomes like of different individuals, versus in viruses, there are hundreds of thousands of viruses. They have to be flexible. They should not be rigid about patterns that, okay, I'll allow only 2 mismatches, or the gap sequence can only be 3 letters or 50 letters long. We want the gap sequences to be, without constraint. So this is based on toolkit that I developed as my very first exercise in computational biology way back when I was a student. And I still am a major Harry Potter fan. So this was named Paternix Revelio, like the spell Specialis Revelio in Harry Potter. Of course, the name did not catch on. even in my own group. So I just call it Biological Language Modeling Toolkit now. So I developed a toolkit that does the pre-processing of genome sequence into suffix array, rank array, and longest common prefix array. And using this data structure, there are several tools that are developed on top of that data structure. So the suffix array is basically this. If you have a long genome sequence and you number every position in the genome, so let's call the suffix, let's say suffix at position 8, right? So it's the letter, it's a sequence that starts at position 8 and goes all the way to the end. Now, let's say there's a suffix at position 20. It's a sequence that starts at 20 and goes all the way to the end. So if you consider every possible suffix in the genome, and then you arrange it lexicographically, there are algorithms to do this, and you can do this in linear time. And the space also it's efficient. It can be efficiently stored because you don't have to store these repeating sequences again and again and again and again. So you have one sequence representation, and you're simply swapping the positions to figure out that, hey, you arrange them in this order of positions, 8, 16, 31, 7, 15, then their corresponding suffixes will be in lexicographic order. It's very useful because if you do this, then all the patterns that are repeating will occur one after the other. So we use this extensively in both finding palindromes. That is, we define palindromes. We start with 8 bases as the basic one, but then we extend it as long as it continues to be palindromic. So we did all of this work. I don't want to get too much into it, but the point is that it does it efficiently. We carried out the analysis. It carries out the palindrome computation for the whole genome in 2 seconds with or without mismatches. And we also found that there are about 4 to 5 palindromes for every 1,000 bases in all chromosomes in human. And then we also studied the variation of these palindromes in 1,000 genomes from the 1,000 Genome Project. So that's published in this paper. Now here we are on to finding out exactly how to find inverted repeats when there is a gap sequence, because we can't generate the seed and then extend it. So what we do here is we make use of the fact that the sequence here in the forward direction, so if you consider this half and this half, it will be the same as the sequence in this half and this half. So what we do is we go and find, is there an occurrence of GACA, that is the forward sequence, howsoever long it may be, anywhere else in the concatenated version of the reverse sequence. So you take the genome, then you concatenate to it the reverse part of the genome, the DNA complement read in the reverse order. And then you take the full sequence and then do the suffix area analysis and see whether you find a sequence that appears in the first half that also appears in the second half. If they do, that means the positions corresponding to the second half in the original genome would be the complement of the first part of the genome. It's a very neat idea. It works really great because it churns out like inverted repeats in a matter of a couple of seconds. So we analyzed, this is a pilot project, we analyzed 13,000 fully sequenced viral genomes from NCBI, and then with the BLM toolkit, and then wrote some scripts to analyze the output of BLMT on the 13,000 genomes. Comparison, we first checked that BLMT is able to detect more palindromes than other tools that exist that have some constraints of parameters. We sort of characterized how many viruses exist with different density of number of IRs. So this is, for example, 3 IRs per 10,000 genomes, so very low density. There are 2,000-ish genomes. How about 6 inverted repeats per 10,000? There are 2,500 of them. But if you take even high density, like 48 inverted repeats per 10,000, relatively high density, there are still about 500 genomes, viruses that have that many inverted repeats. How about if we separate the viruses into whether they infect the bacteria or human or other vertebrates and so on? So we find that this is the number of IRs per kb. So the density for all of these seems to be somewhere between 5 to, let's say, 10. 5 to 10 IRs per 10,000 bases. We are working on this, analyzing it, and we hope to work with biologists, including our collaborator, Professor Matthew Schraman, and we'll take it forward and hopefully present it again. And so all of these are undergrad students. Everyone look at this. This is from Philippines. I have not even met him. I only interacted with him on internet. Same here, Madhura Sen is from India. I have not met her. This student I met because he's from CMU Qatar, Jingshan Ga, George Rivera, Madhura Sen, and Professor Matthew Schraman. And I'm Madhavi. Thank you.